Okay, so let's talk a little bit more about transcription. And again, transcription is the process of taking our DNA and converting it into our messenger RNA. A couple things are needed. We need some transcription factors that are actually going to activate the process of transcription because they're going to go in there and help loosen up those histones. Remember that our DNA double helix is wound around that complex of 8 histones pretty tightly, so we have to loosen it and so that we can get our enzymes in there to undergo transcription. We also have to have a promoter come in and bind. Again, the specifics of all this that's in your test book are not important. I just want you to have a general understanding of what transcription is. So we need transcription factors. Again, they come in and loosen up the DNA from those histones so that we can access it. We have to have a promoter that binds the DNA. So transcription factors will come in and bind that promoter to initiate transcription. And we need an enzyme. And our enzyme is RNA polymerase. So we talked about for replication, we needed an enzyme that was responsible for bringing in the nucleotides to synthesize the new DNA strand. So that's DNA polymerase. When we're talking about transcription, we need an enzyme to come in and lay down the nucleotides again, but now it's an RNA polymerase. Again, just really get a general idea. If you take principles of biology, you will have to know these steps in a lot more detail. But again, I just want you to have a general understanding. So both transcription and translation have the 3 basic steps: initiation, elongation, and termination. However, what's going on in each one of these steps is a little bit different depending on whether or not we're talking about transcription or translation. So for transcription, again, our enzyme is RNA polymerase. So that's what's going to begin initiation. So RNA polymerase comes in and it's going to separate the DNA double helix into 2 strands. It's also going to function by adding complementary nucleotides so that we can build our RNA molecule. As we're building it, the RNA molecule is hybridized to the DNA, so it forms what's called a DNA-RNA hybrid. And then basically it's going to keep continuing to transcribe until it reaches a segment on the DNA called a termination signal. Once it reaches a termination signal, the RNA polymerase is simply just going to follow up the DNA and stop transcription. And that's the point at which we have our completed mRNA. So again, transcription, we're taking DNA, we're converting it into an mRNA molecule. The enzyme responsible for that is RNA polymerase. It's going to start by binding to a promoter region, and it's going to stop transcribing when it hits a termination signal. So like I mentioned previously, the first RNA that's produced is what's called a pre-mRNA because it still contains that junk and it has to undergo some processing. So what's going to happen is it's going to form something called a spliceosome that's going to go in there and basically cut out all the junk. The junk that it cuts out is called introns. The important regions, the actual coding regions within the DNA, those are called exons. So once we cut out all the introns or the junk, we're left with these exons, and we need a spliceosome to come in there and splice these all together so that we have our functional mRNA. So now we have our functional mRNA, and we have to take our mRNA and convert it into a protein. So when we went from DNA to RNA, both of those are made up of nucleotides. So it's a pretty one-to-one process. Here's a nucleotide, complementary base pair to another nucleotide in order to make our mRNA. Now we're taking our mRNA, which is made up of nucleotides, and trying to convert it into something that's a completely different language, which would be amino acids that make up proteins. So because of that, and because we have 20 amino acids and we only have 4 different possible nucleotides, we have to have some other way to go in and build these polypeptides. Again, 20 amino acids, if we only have 4 nucleotides, how do we code for that many? Typically, you would think with 4 nucleotides, 4 amino acids. So they figured out later that in order to code for 20 different amino acids, there was a code that was read by more than one nucleotide. So how these nucleotides on the mRNA are read are in sets of 3, or also called triplets or codons. So that's what the genetic code is showing you. This right here is the genetic code. Basically what it's doing is it's showing you all the possible triplets or the codons of nucleotides. We know that this is showing us 3 nucleotides on an RNA molecule because we see uracil. Remember, uracil is not in DNA, it's only in RNA. So if an mRNA molecule comes across 3 U's in a row, 3 uracils in a row, that means that our TNA molecule is going to come in and lay down a phenylalanine in that place, which is an amino acid. And it's going to continue to read the mRNA in sets of 3, or these codons, until we basically run out of mRNA to read. So we read 3 nucleotides, lay down an amino acid, go to the next set of 3 nucleotides, lay down another amino acid, and so on and so forth until we have our completed polypeptide. Looking at this genetic code over here, there are 64 possible codons, again, those triplets of 3. However, only 61 actually code for an amino acid. So these are your amino acids over here. Over here, if you look, these 3 do not. When the ribosome comes across one of these 3 codons, so either UAA, UGA, or UAG, it's not going to come in and lay down a amino acid. Instead, it's going to be a signal for it to stop. The ribosome is going to disassociate and fall off the mRNA, and translation is going to be completed. Also, if you notice and look at the genetic code, we can see just right here that 4 different codons code for the same amino acid. So we also say that the genetic code is redundant, and this is going to help protect against transcription errors. So again, when we talked about transcription, going from DNA to mRNA, we started with a promoter sequence, at which point our RNA polymerase bound. We finished transcription when we hit the termination sequence. When we're talking about translation, we start when we hit a specific codon, which is always going to be AUG, and we stop translation when we hit one of these 3 stop codons over here. That's when our ribosome is going to come in and disassociate. Another thing I want to point out while we're looking at this genetic code is the start codon does code for an amino acid, and that amino acid is methionine. So if we come in and we don't start translation until we hit AUG, and because AUG codes for methionine, that means every single protein that's ever made, the first amino acid in that protein sequence is going to be methionine, because that is the start codon. I do want to say that this methionine can later be cleaved off, but initially, like I said, the first amino acid in every polypeptide is going to be methionine. So we've already talked about the ribosome and it being the site of translation. So that's where our mRNA strain is going to come in and basically land. But we didn't really talk about what's going to bring in all of these 20 different amino acids to the ribosome itself so that we can build our polypeptide. And what's going to accomplish that is this tRNA molecule. And the tRNA molecule is represented here in blue, and it has something that's called an anticodon at one point, and it carries the amino acid on the other point. So the anticodon is not the codon sequence, it's actually the complement to the codon sequence. And we're going to talk more about that in a little bit. So TNA knows what amino acid to come in and lay down by coming and binding its anticodon to the codon so that it knows it's supposed to lay down, in this case, an isoleucine. Again, I'm just going to go quickly. We have the same 3 steps that we had in transcription, just a little bit different things are happening in each of the processes. So we still have initiation, elongation, and termination. So when we talk about initiation, again, we're not working with an enzyme like RNA polymerase. Instead, we're working with our ribosome. Our ribosome is made up of 2 different subunits. One is what we refer to as a small subunit, and then one is the large subunit. Collectively, they come together and form the complete ribosome. So the first thing that happens is that your mRNA molecule is going to come in and be there. The small subunit is going to attach to the mRNA molecule and basically scan it until it comes across that start codon, which is AUG. So once it comes in and it scans and it finds the start codon, which is AUG, a special tRNA that we call an initiator tRNA comes in and lays down a methionine at that point. Again, how does it know to come in and lay down? Because it's going to bind with its anticodon to this codon right here and then place down the methionine. Once the anticodon of the initiator tRNA binds to that start codon, the large ribosomal subunit is going to come in and finally attach to the small one. So we have our complete ribosome product. I'm going to show you a picture in a little bit that'll make more sense. Then we move on to the next phase. After we've laid down methionine and found our start codon, we can actually go in and elongate our polypeptide by coming in and laying down more amino acids as we recognize these triplets of nucleotides. So elongation has 3 substeps. It's going to be where we come in and the tRNA recognizes the codon. Once we lay down our amino acid, we have to join that amino acid to the previous amino acid that was laid down. And how do we do that? We create a bond between the 2 amino acids. And the bond that's created is referred to as a peptide bond, which is why proteins are also called polypeptides, because they have multiple peptide bonds between their amino acids. The third subphase of elongation is translocation. So once we form that bond, we have to kind of shift it from one site to the next site so we can bring in another tRNA that's carrying another amino acid. You don't need to worry about the specifics or the names of the site. Again, just kind of understand the general process of elongation when we're talking about translation. Another thing to think about is a lot of times textbooks, or if you've learned this before, you kind of focus solely in on one ribosome going in and translating one mRNA. In reality, we don't just have one ribosome attached to an mRNA molecule. We actually have multiple ribosomes going in and translating this mRNA into multiple different polypeptides at the same time. And when we have multiple ribosomes attached to an mRNA, we then call it a polyribosome. So here's an example of our mRNA structure right here, that this blue thread-like structure. We can see we have several ribosomes bound at various stages of the translation process attached, and like this one just finished translation. Because that one finished, we have another one coming in. If we didn't have these polyribosomes coming in and doing multiple translational processes on the same mRNA, we would not be able to produce enough proteins to survive. So this is actually a really important process that we have, where we have multiple ribosomes bound to the same mRNA, so we can produce enough proteins in order for us to sustain all the needs in our body. So the final step is going to be termination. And again, I'm not really worried about the site, but the big thing here is that when our ribosome reaches the point where it's reading one of those 3 stop codons, that's when our ribosome is going to disassociate, and actually water is going to be added in place of an amino acid. And the binding of the water is actually what's going to break it and cause everything to kind of fall apart afterwards. So here's the big picture. These are the sites I was talking about. Again, not important. The big thing here is that we won't start bringing in, our TNA will not start bringing in amino acids until we scan and we find that AUG start codon. Our tRNA is going to complementary base pair using its anticodon, which is right here, and base pair to the codon that's present there. Once it does that, it can drop down its amino acid, and then this whole structure is going to shift to the left so that we can have another codon enter into the correct part. So we can see here, now we've moved on. Now we're laying down, and now we have several polypeptides going right there, and we just kind of have this shifting going on. So once we've done laying down an amino acid to this growing polypeptide chain, it will shift over. The empty tRNA will be released, and a new codon is going to enter into a site, at which point another tRNA is going to bring in another amino acid. It's just kind of this shifting and rotating process. Again, do not worry about the site names or in which direction it shifts. Just have a general understanding is that a tRNA molecule is bringing in a amino acid. It's going to complementary base pair using its anticodon to the codon on the mRNA, and then it's going to form a peptide bond, and then it's going to shift over and kind of leave. And that's going to continue until it hits one of those three stop codons. I took this off of the exam, so you do not need to worry about this on this first exam. And I'm going to make a separate video on this.